स्टूडेंट, जैसा कि हम बात कर चुके हैं कि हमें सेल लाइजेट की ज़रूरत पेश आएगी या टिश्यू लाइजेट की ज़रूरत पेश आएगी टू एक्सट्रैक्ट द आरएनए। इससे पहले के जो 3 मॉड्यूल थे उसमें हमने सोर्स ऑफ सैंपल और उसकी प्रोसेसिंग के हवाले से बात की थी, हाउ टू गेट द टिश्यू लाइजेट और हाउ टू गेट द सेल लाइजेट। नाउ वी आर गोइंग टू टॉक in detail about the process to get the RNA purified from the cell lysate। यहाँ पे मैं स्टेप 2 से इसलिए स्टार्ट कर रहा हूँ कि हमने जो सेल लाइजेट या टिश्यू लाइजेट को प्रिपेयर किया है, दैट वाज आवर स्टेप नंबर 1। सो इन दिस मॉड्यूल वी आर गोइंग टू टॉक अबाउट स्टेप नंबर 2 एंड सब्सिक्वेंट स्टेप्स। Once the lysate is ready, we have to transfer it to a polypropylene tube and we have to add sodium acetate, phenol, chloroform, isoamyl alcohol in the solution D। अगर आपको याद है जो हमने लाइजेट प्रिपेयर किया है, that is prepared in solution D। Now keeping in view the volume of the solution D, we add these buffers sodium acetate, phenol, chloroform, isoamyl as per their required concentration in the required or used volume of solution D। Once these buffers are added into the solution D, cap the tubes and vertex them vigorously so that the homogenate overall it gets mixed. This vigorous vertex mixing should be done for 10 seconds. After 10 seconds, leave the tube on ice for 15 minutes. Now these 15 minutes are the time when we will see maximum or complete dissociation of nucleoprotein complex। जब हम बात करते हैं सेल की, सेल में साइटोप्लाज्म की, प्रोटीन्स की, न्यूक्लिक एसिड्स की, everything is interacting with each other। So we want RNA should be separated from any kind of protein if it is associated। So this step is going to cause complete dissociation of nucleoprotein complex। उसके बाद नेक्स्ट स्टेप आएगा जिसमें हमें सेंट्रीफ्यूगेशन की तरफ जाना है 10,000 G के ऊपर फॉर 20 मिनट्स एट 4 डिग्री सेंटीग्रेड। द पर्पज ऑफ दिस सेंट्रीफ्यूगेशन इज टू सेपरेट द एक्वस अपर फेज which contains the RNA from rest of the cellular debris or unwanted biomolecules। So at this stage, the step number 4, the centrifugation is going to separate the two phases. The upper aqueous phase is going to contain the RNA which will be used for further processing। Once the centrifugation is done, then we are going to add equal volume of isopropanol in the extracted RNA। Equal volume, if you have 2 milliliter of the upper aqueous layer, then add 2 milliliter of the isopropanol। Mix it well, allow the RNA to precipitate for 1 hour or more at minus 20 degrees centigrade। This is a crucial step। If we don't follow the times and the temperature, the RNA will not precipitate to its maximum, and at the end the concentration of RNA in the RNA solution will not be sufficient enough to proceed further। So we have to take care of this precipitation step। Once RNA is precipitated, we collect this precipitated RNA by centrifugation at 10,000 G for 30 minutes at 4 degrees centigrade। Isopropanol is no more required। RNA is precipitated at the bottom, isopropanol is no more required। We are going to decant or discard the isopropanol। We have left with the pellet of RNA। This RNA pellet is dissolved again in solution D with a volume of 0.3 milliliter for every 1 milliliter of solution D which was used in step number 1। In other words, we are saying that at this stage we are reducing the volume and we are only using 0.3 ml in place of 1 ml of solution D। We have to transfer all this solution to a microfuge tube, vertex is well, and precipitation of RNA is done once again with 1 volume of isopropanol at minus 20 degrees centigrade for 1 hour। This step is going to be repeated for precipitation। This precipitated RNA is collected by centrifugation for 10 minutes at 4 degrees centigrade। Once this pellet is achieved, again after centrifugation, RNA settles at the bottom in the form of pellet. Upper solution or the supernatant is no more required, is discarded or decanted. Then we have to wash the pellet. The washing of the pellet is done by 75% ethanol. 2 times 75% ethanol is used to wash this pellet, and every time we centrifuge to get our RNA settled down and to leave behind the ethanol which is aspirated or which is decanted easily. The next step is don't leave any traces of ethanol, because ethanol, if it is left behind, is going to interfere with the RNA concentration estimation or may cause the problem with the RNA during the storage. So what we have to do, using a pipette tip, we can remove any minute or little smaller drops of alcohol, and subsequent ethanol removal can be ensured by leaving the tubes open on the working bench, so that because of evaporation the alcohol or ethanol should be evaporated completely, and we should not have any kind of ethanol associated with the RNA pellet. Once the ethanol is removed, now this RNA pellet can be dissolved in DEPC treated water. If you remember, हमने बात की थी कि RNases are very aggressive, they are very active, they are found everywhere. So whatever we use should be treated with DEPC. So now at this step, because we are going to dissolve the RNA pellet into water, this water should be DEPC treated so that there should not be any RNAs, and the RNA should not be digested by these enzymes. After making solution of RNA or dissolving the RNA into the DEPC treated water, we can store this RNA at -70 degrees centigrade for subsequent usage. This next step after isolation is to determine the concentration of RNA. As this is nucleic acid, we can determine its concentration at 260 nanometer and 280 nanometer, and then finding the ratio of absorbance at 260 and 280 nanometer, we can find out the purity level of the RNA. How pure is the RNA? जैसा कि मैं पहले बता चुका हूं कि RNA जो प्योरीफाइड है उसका जो रेशियो है एब्जॉर्बेंस की 260 एंड 280 इज इन बिटवीन 1.8 to 2.